Hello and welcome everybody to my channel Omini Bio Entrance, and today we will be discussing National Entrance Screening Test NEST for 2021 session 2 question paper. I will be discussing only the biology section. If you are new to my channel, please do like, share and subscribe. So coming to the first question. Some characteristic features, that is presence is given by plus and absence is given by minus, of certain organisms belonging to various categories P, Q, R, S, T are listed in the table. The category that best satisfy the option. So membrane bound organelles, they are found only in Q, R and S but not in P and T. Then introns are found only in Q and R and S. So naturally they are eukaryotic, whereas P and T are prokaryotic. Then cell wall is present in P, Q, R and S but not, and also in T. Then flagella or cilia is present in P and Q only, but R and S and T do not have flagella. Then chlorophyll A is present only in Q and S, so they are autotrophic. And T is not having chlorophyll A, but still it is autotrophic. So mode of nutrition of P is heterotrophic, Q is autotrophic, R is heterotrophic, S is autotrophic and T is also autotrophic. So P will be Monera. It is does not have introns, so it is prokaryotic. Then flagella and cilia are present and the mode of nutrition is heterotrophic, so naturally it is Monera. Whereas T is cyanobacteria, because even though it is not having chlorophyll A, it is autotrophic and it has got cell wall also but no cilia and flagella, so T is naturally going to be cyanobacteria. Then we will see the others. Q is autotrophic and it is protista because it is having cilia, flagella, cell wall, introns, membrane bound organelles and chlorophyll A, so it is autotrophic and naturally it is protista. Then R is heterotrophic and no chlorophyll and flagella are present, so it is naturally a fungi. So R is fungi and S is autotrophic with chlorophyll A being present and no flagella and cilia, so it is plantae. Answer is option S is plantae. Answer is option 4. Then next one. A double-stranded DNA template with the sequence. So they have given the sequence. Was PCR amplified using forward and reverse primers? Given the size of the sequence of the new PCR fragment product from the table, the correct combination from the options is. So 1. They have given us fragment length is 30 and it matches with K. That is the DNA fragment sequence. So we will see how it is done. So here you have got the sequence of where they have given in the 5 prime 3 prime direction. So this is the particular sequence that need to be amplified. So you know the 3 prime 5 prime sequence that is complementary to the 5 prime 3 prime sequence. So this is the 3 prime 5 prime sequence. Now there is the forward primer. It is given as GAACTA. How did you get the sequence of this forward primer? Because the forward primer will bind to the 3 prime 5 prime complementary strand and it will synthesize the DNA in the 5 prime 3 prime direction. Why? Because DNA polymerase can add nucleotides only to the 3 prime end of the DNA of the primer. So the forward primer, that is 5 prime 3 prime, it will be GAACTA and it is the same sequence as this particular sequence strand because it is complementary, so naturally it will be same as the one given for this particular strand. Then you have got the reverse primer, that is 5 prime to 3 primes, so in this direction. So it is ACGTAG, that is the reverse primer. So this is in the 5 prime to 3 prime direction. So it will be complement or reverse complement of this particular first strand. Now these two primers, they will bind to the target region of the DNA, that is the complementary region where it can form complementary base pairs. Then it will synthesize the DNA template. So what is the size of the fragment? That is, it is having how many nucleotides? So from this here, from this to this, it is 30 nucleotides. So from here, this region to this region, it is 30 nucleotides. Similarly, from this region to this region, it is 30 nucleotides. So 30 nucleotides is correct. Then this is the particular sequence that will be amplified because you have created the primers for that particular region. So this particular DNA sequence will be amplified. So option, I don't remember the option, but 1 and K is correct. So next question. An angiosperm plant of genotype this pollinates. So that is the male parent because it pollinates, they have given. Another plant with genotype this. So the another plant is the female parent. If the genes are not linked, the genotype of the embryo and endosperm in the resulting seeds will be. So I have shown it. So this is the genotype of the male parent, that is the one which is pollinating this particular parent. So this is the female parent. So it will produce 2 sperms. Any of them can cross with this particular parent and it will produce only 1 egg cell. And the other will be the fusion of polar nuclei resulting in the formation of endosperm cell upon fusion with the male gamete. Okay, so now upon fusion of this particular DNA sequence with this particular DNA sequence, you will get capital P, small p, capital Q, small q, capital R, capital S, small s. Now there is the diploid central cell which upon fusion with the male gamete that will give rise to the endosperm. So the diploid central cell will have the same sequence as that of the parent. It is diploid central cell. So this will be the same sequence as that of the parent and that will cross with small p, small q, capital R and S and you will get a 3N or triploid number for endosperm. So the answer here is option, wait, 3. Okay. Here are the options. Now if you see for this particular genotype, so the option 3 is the answer. Now capital P, so you will not get capital P if you see the cross because the male parent is p, small p and the female parent is capital P. So upon crossing with the male parent you will get only capital P, small p genotype, not capital P genotype. So 1 and 4 can be eliminated whereas you will see for 2 and 3. So in case of 2 and 3 the genotype of the embryo is the same. Whereas if you see the endosperm, you will see that here also they have got one capital P written over here. That will not happen because the male parent is having only small p. So it will fuse with the female parent, it will result in having one, it is endosperm, so it will have 2 capital P and 1 small p. So naturally option 3 is the correct answer. Then next one. In 4 separate experiments that is given in the table here, a vector DNA VXYZ, that is 6000 base pair, that is this is the vector DNA, and a DNA X, that is 1500 base pair fragment was digested with the indicated restriction enzyme. So you have got the vector DNA, you have got the gene of interest, that is DNA X. It was restriction digested with these particular enzyme combinations. Then both the digested vector and DNA X were purified and ligated. Then you purified the vector, then you purified the DNA also after digestion. Then both were, after restriction, and they were purified and ligated. Ampicillin resistance represent the ampicillin resistant gene and ORI is the origin of replication. So here is the ampicillin resistance gene and here is the ORI. The resultant recombinant vector was transformed into Escherichia coli and incubated in a liquid nutrient medium with ampicillin for 24 hours at 37 degrees Celsius. Based on the restriction enzyme used for cloning, choose the correct option. So, first option. In experiment 4, that is over here, experiment number 4, you are digesting with KPN1 and SAL1. So in experiment number 4, the transformed Escherichia coli will be ampicillin sensitive and the recombinant vector cannot be isolated from these cells. So in option 3, if you see, it is KPN1 and SAL1 digestion. So it will cut over here, KPN1, SAL1. Similarly here, KPN1, SAL1. It will cut over here. It doesn't cut the HIN3 region. It doesn't cut the HIN3 region. Since HIN3 region is not restricted, it will be ampicillin resistant and not ampicillin sensitive. So first option is ruled out. Then next one. Next one is experiment. Okay, option 3 we will see. In experiment 1, that is 1, it is digested with BAM-H1 and SAL1. So it has cut the ORI. So BAM-H1 is digested, then SAL1 is digested over here. Then BAM-H1 and SAL1 for this particular DNA also it is digested and it is ligated. Now the ORI is cut. So in experiment 1, the transformed E. coli cell will be ampicillin resistant. Yes, because HIN3 region is not cut, so ampicillin resistance will be there. And the recombinant vector can be isolated from these cells. You cannot isolate the recombinant vector because the recombinant vector will not be able to multiply and cannot be isolated. Why? Because the ORI is restricted. So origin of replication itself is restricted, so naturally recombinant vector will not be able to multiply. Then coming to option 4. In experiment 3, the transformed Escherichia coli. So experiment 3 is KPN1 and HIN3. So over here, KPN1, HIN3, KPN1, HIN3. So if you digest over here, so HIN3 region is digested. So ampicillin resistance will not be there, it will be sensitive. So transformed Escherichia coli will be ampicillin sensitive, yes. And the recombinant vector can be isolated from these cells. Okay. Now your ampicillin sensitive. So once ampicillin sensitive, selection of recombinants will not be possible on ampicillin medium as both recombinants and non-recombinants. So both recombinants as well as non-recombinants. Non-recombinants will grow. So you cannot isolate the recombinant vector. So option 4 is also not correct. Then coming to option 2. In experiment 2, that is EcoR1 SAL1, that is over here, EcoR1 SAL1, EcoR1 SAL1. The transformed Escherichia coli will be ampicillin resistant. Yes, HIN3 is not damaged, so it will be ampicillin resistant. And the recombinant vector can be isolated from these cells. Yes, you can even isolate recombinant vector from this particular restriction digestion. So experiment 2 is correct. Then next one. DNA fingerprinting was performed on human skeleton S to identify a missing person reported by 4 different families. That is F1, this is first family, this is F2, this is F3 and this is F4. 4 short tandem repeat, that is STR loci, were independently PCR amplified using DNA isolated from the family members and S also. The diagram represents PCR products after gel electrophoresis showing the 4 STR loci, that is red, blue, green, yellow, black. Green is not there, okay, red, blue, yellow and black colored lines. Based on these results, the family to which S relates. So from here how you have to compare is all the bands in S should match with either mother or father. Okay. Only then you can say that the S is son of or daughter of this particular mother and father. So answer here is option F3, that is family F3. So red matches here with the father. So blue line from S matches with the mother. This yellow line matches with the mother as well as the father. Then the black line, it matches with the father. Red line matches with the mother. Blue line matches with the father. And black line matches with the mother. So S is the son or daughter of this particular mother and father of family F3. So 1 is F3. While if you see this, so S is having a red band over here, it matches with the mother. But blue band doesn't match with mother or father. Okay, blue band is there but it doesn't match with mother or father. So this S cannot be the son or daughter of this particular family. Similarly here also a red band is there but it doesn't match with the father. Okay. Similarly a red band is here, it doesn't match with mother or father. So all these conditions will say that F2 in family F2 S does not belong. It's not the father, daughter or son of this particular mother and father. Then family F4 if you take, you can see a red band matches with mother and father. Blue band doesn't match with mother and father. Then you have got the yellow band that is matching. Black is also matching. Red does not match. Here also black is not, okay, black is present, blue is present. Here red is not matching with mother and father. So S cannot be the son or daughter of this particular mother and father of family F4. So he or she, that is S, is the mother, son or daughter of this particular mother and father of family F3. So answer is F3. Then next one. Passiflora plants produce toxic glucosides that protect its leaves from several insects. However, heliconius caterpillars sequester these compounds for their own benefit. Thereby, it gains access to food that few insects can eat. Moreover, Passiflora leaves produce yellow spots that resemble heliconius eggs. So heliconius butterflies avoid laying eggs on these leaves that already have eggs or yellow spots, ensuring hatching of fewer caterpillars per leaf. Thus, Passiflora with these yellow spots are less likely to be eaten. In addition, Passiflora secretes abundant nectar that attract ants responsible for caterpillar mortality. Given these interactions, the correct option is. So option 1. Mutualistic relationship exists between Passiflora and heliconius caterpillars. Not mutualistic as Passiflora safeguard its leaves, secrete nectar that attract ants who feed on the caterpillars. So it is not mutualistic attraction. Then the interaction between Passiflora and heliconius caterpillars is commensalism. When one is benefited and the other is not harmed nor benefited, it is called as commensalism. Here neither is either benefited but both are harmed. So the interaction between Passiflora and heliconius caterpillars is not of commensalism. Because Passiflora, it produces yellow spots that resemble heliconius eggs, so it will prevent butterflies from laying more eggs on the leaves. So less caterpillars are produced, hence Passiflora leaves are safeguarded. It also produces toxic glucosides which heliconius caterpillars use as food. But at the same time, Passiflora secretes nectar that attract ants which causes caterpillar mortality. So neither is either benefited but both are harmed. So it is not a case of commensalism. Then an invasive glucoside resistant ant population will not affect heliconius members. It will affect heliconius numbers. So glucoside resistant. So glucoside is secreted by Passiflora to ward off insects and protect its leaves. But if ant population that is resistant to this glucoside, it will affect heliconius members because ants will naturally eat heliconius caterpillars. Then this is a typical example of coevolution between Passiflora and heliconius. So coevolution is the process of reciprocal evolutionary change that occurs between pair of species or among groups of species as they interact with one another. The activity of each species that participate in the interaction applies selection pressure on the others. So Passiflora and heliconius, it is a kind of co-evolution. So answer is option 4. Then next one. Sterilization is the process in which all living organisms including microbial endospores are destroyed. A disinfectant is an antimicrobial agent used on inanimate objects such as instruments. An antiseptic is an antimicrobial agent used on the skin externally. Antibiotics are administered either topically, orally, intravenously and can cause side effects. Based on this information, the correct option is, for second option, antibiotics work well on bacteria, fungi and viruses. You cannot say that from the given data. Okay, then next one. In contrast to antiseptic, an antibiotic is specific to a single species of microorganism. Antibiotics are broad spectrum and are not specific to one particular single species. So that is also wrong. Disinfectants and antiseptics completely destroy all forms of life. No. There may be certain ones which are resistant to this particular disinfectant or antiseptic. Then first option, viruses are destroyed by sterilization of inanimate objects. So viruses are destroyed by sterilization of inanimate objects and not by antibiotics. Antibiotics do not infect viruses. So option 1 is correct. Then next one. The family tree of lemurs split off from the last common primate ancestor as wet-nosed monkeys. So, while the other branch of dry-nosed monkeys eventually gave rise to the human lineage. So, there was something called as the last common primate ancestor, from where lemurs and, that is, wet-nosed monkeys and dry-nosed monkeys split off. The lemurs were the wet-nosed monkeys and the dry-nosed monkeys were the humans. So, that much clear. Thus, lemurs are ancient evolutionary cousins of humans, of course. While humans entirely depend on vitamin C as a dietary supplement, that is citrus fruit, lemurs can synthesize their own. So, humans cannot synthesize vitamin C, they have to take it from their dietary supplement, that is eating citrus fruit. So, this is an example of divergent evolution wherein lemurs have retained gene P. So, divergent evolution is where it has arose from the single common ancestor, has given rise to two, or I can say a single common ancestor has given rise to two different species. And the difference between these species is that lemurs have retained the gene P for vitamin biosynthesis, whereas humans cannot synthesize vitamin C. Then one of those responsible for the option that best satisfies the information is. So option 2, if you see, they have given horizontal gene transfer of gene P from DNA of dietary citrus fruit conferred vitamin C independence in lemurs. So horizontal gene transfer or lateral gene transfer is the movement of genetic material between unicellular and or multicellular organism other than by the transmission of DNA from parent to offspring. So it is said in the question that it is an example of divergent evolution where lemurs retained the gene P from the ancestor and humans lost it. So option 2 is not correct. Then dry-nosed monkeys do not require vitamin C and therefore have lost the gene P. So it is said that dry-nosed monkeys gave rise to humans and humans entirely depend on vitamin C as a dietary supplement. So dry-nosed monkeys do not require vitamin C is not correct. They require vitamin C but they take it as a dietary supplement and therefore have lost the gene P. So 3 is also wrong. Then consumption of citrus fruit caused mutations in human gene P leading to its loss of function. So consumption of citrus fruit did not cause any mutations because lack of gene P, humans are consuming citrus fruit. And consumption of citrus fruit did not cause any mutations in human gene P. So during evolution itself, it was lost. So option 4 is also not correct. Then option 1, humans lost the promoter but retained the rest of the protein coding features of the sequence of gene P. That can be correct since promoter is not present or it is lost or it is mutated, it can result in no protein being synthesized by the gene P for vitamin biosynthesis. Okay. So answer is option 1. Then, during an excursion, students found a non-woody flowering plant and wanted to classify it either as monocotyledons or dicotyledon. The option that will not help them in classifying the plant is, so root architecture, one is having taproot, one is having fibrous root. So monocotyledons and dicotyledons you can classify based on root structure. Then number of cotyledons, one is having single cotyledon, the other is having two cotyledons. So based on the number of cotyledons also, you can classify either as monocotyledon or dicotyledon. Then vascular bundle arrangement, one is exarch, one is endarch, one is polyarch whereas other is not. So based on the vascular bundle arrangement also, monocotyledons and dicotyledonous plants can be distinguished. So what is the option that will not help in classifying the plant? It is the shape of the flower. So this is a direct question. Then next one. Oxidative phosphorylation is carried out by electron transport chain present in the mitochondria of aerobes. The NADH or FADH2 produced in TCA cycle transfers their electron to the electron transport chain to finally reduce oxygen. Based on this information, the correct option is complex 1 of electron transport chain is also an enzyme of TCA cycle. No, complex 1 is NADH dehydrogenase. Then complex 1 transfers electron to complex 2 in the electron transport chain. Complex 1 transfers electrons to ubiquinone, not to complex 2. Then complex 4 is an ATP synthase. Complex 4, it consists of cytochrome A and A3, which is also termed cytochrome oxidase. So it is not an ATP synthase. So 1, 2 and 3 are wrong. Then complex 2 does not transfer proton into the intermembrane space. So complex 2, it does not transfer protons. So that is correct into the intermembrane space. So option 4 is correct. Then next one. A protein X with isoelectric point 6.5 was isolated. It was prepared in 3 different buffers P, Q and R with pH 4, 6.5 and 8.5 respectively. With respect to the net charge on the protein, the correct option is okay. So isoelectric point is the pH of a solution at which the net charge of the protein becomes 0. So it will have both positive and negative charges. It will be 0. Okay. Then if you take the option Q, it is having pH is equal to Pi, that is 6.5 is equal to the isoelectric point 6.5. So Q is having a net charge of 0. Or else we can say that both positive and negative charges are present in Q. So Q is option 4 if you see, while both positive and negative charges are present in Q. So that is correct. Now we will see for the net charge in P. Now pH is less than Pi. So what is the pH of P? It is 4. 4 is less than Pi, that is 6.5. So 4 is less than 6.5. So it will carry a positive charge. So the net charge in P is positive. Then if you take R, R is having a pH value greater than Pi. That is 8.5 is greater than 6.5. So it will be negative charge. So the option is option 4. Net charge in P is positive and is Q and in R is negative, while both positive and negative charges are present in Q. So answer is option 4. Then next one. In 2020, Nobel Prize in Chemistry was awarded for the CRISPR-Cas9 system. This concept was derived from. It was from bacterial defense machinery against a viral infection. It's a direct question. Then next one. In the given pedigree, circles represent females and squares represent males. Filled shapes indicate individuals for a particular trait, while unfilled shapes indicate unaffected individuals. Carrier status is not shown in the pedigree. Study the following statements about the pedigree and choose the correct option. So they have given some statements for this particular pedigree analysis. So coming to the analysis of this particular pedigree. So as I have, Males and females equally likely to transmit the trait to their children. So here it is the female that is transmitting to one male and one female offspring. Okay, so it is in contrast with the sex-linked dominant or recessive inheritance. So X-linked recessive inheritance need not be taken into consideration over here. So 1 and 2 statements we need not have to take into consideration. Then no difference in expression between male and female, that I told you. Already I told you. Then mating between affected individual produce 100% affected offspring. So if there is mating between affected individuals, that is these two, if there is a mating between 3 and 4 over here, then you will get 100% affected offsprings. Then unaffected parents can produce affected offspring, that is in heterozygous condition. So if this parent is heterozygous, it is producing offsprings. Okay, so this is 2 and 3 are being produced. Now this can skip generation. So it is present in this generation but not present in this generation. Okay, but here it is not present in the parents but it is present in the offspring. So it can skip generation. You can only express the trait if you are homozygous. Only homozygous. So from here itself you can make out that this particular trait is expressed only in homozygous condition. So all these show that it is option 3, that is it follows autosomal recessive mode of inheritance. Now for the other statements you have to see the pedigree. Okay, I have drawn it in the next slide. So these are the genotypes of the parents and the offspring for that particular pedigree. So here capital P small p and capital P small p has to be present, that is they should be carriers or heterozygous, only then this particular offspring will be a diseased offspring, that is carrier of the disease, autosomal disease. So 2 will be, only then it will be carrying the disease. So we will see the statements. Individual 2-1 is a carrier for the trait. So this is the individual 2-1. This is the individual. It is to be a carrier of the trait. Only then it will give rise to homozygous recessive condition. That is small p, and it will be affected. So either only because this is a carrier for the trait, 2 and 3 have got the autosomal recessive genotype. So that is correct. Then individual 3-4 is a carrier for the trait. So fifth statement is individual 3-4, that is this particular trait is a carrier for the trait. Only because it is carrier, that is 5 is also a carrier, 4 is also a carrier. That is why you got the offsprings 3 and 4 as autosomal recessive mutants or homozygous recessive genotype. So option statement 5 is also correct. Then individual 1-1, that is this particular individual is a carrier for the trait, that I already explained. If it is not a carrier, then this particular phenotype would not genotype would not have been obtained. So statements 3, 5, 4, 5 and 6 are correct actually, but in the option they have given only statement 3 and 5 combination, which is correct. So option 4 is the correct answer. Then a researcher determined the initial velocity of an enzyme in 3 different reactions corresponding to lines P, Q and R. So these are the lines in the graph with increasing substrate concentration. This is increasing substrate concentration, and this is a Lineweaver-Burk plot because they have taken reciprocals, 1 by concentration of S, 1 by V0. Okay, the results were analyzed using reciprocal plot where 1 by V0 is plotted against 1 by S graph. Reaction 1 was performed without any inhibitory compound. Okay. Reaction 2 was performed with compound X that inhibits enzyme activity by binding to the substrate binding site. And reaction 3 was performed with compound Y that inhibit enzyme activity by binding to the site other than the substrate binding site. So naturally, reaction 1, because it is a compound that inhibits enzyme activity by binding to the substrate binding site. It is a case of competitive inhibition. Then reaction 3, it was performed with a compound Y that inhibits enzyme activity by binding to site other than the substrate binding site. So it is not a competitive inhibitor. Then if Km of the enzyme in the reaction 1 is 0.5 millimolar, then the correct statement from the following options is or are. So they have given the Km value as 0.5. So minus 1 by Km is equal to minus 2. So this value in the graph which gives a Km value of 0.5. So if you see that minus 2, that is minus 1 by Km, you have got minus 1 by 0.5 is equal to minus 2. So minus 2 corresponds to the value of Km 0.5. Okay, minus 2, this particular minus 2. It corresponds for the Km value 0.5. So this particular reaction, that is R, is reaction 1, that is without any inhibitor. So our reaction 1 is R. Then reaction 2, you will see. What is reaction 2? It was performed with using a enzyme by compound X that inhibit enzyme activity by binding to substrate binding site. So I told you it is the case of a competitive inhibitor. So for competitive inhibitor, Vmax is the same as the normal enzyme, but the Km is larger. So Vmax will be the same, that is over here. So Vmax will be the same, that is P will be the competitive inhibitor. Why? Because Vmax same but Km value increases. So from point minus 2 it has come to point minus 1. So Km value has increased, and reaction, so P is the reaction 2. So reaction 2 is P. So option 2 we can see that reaction 1 is R, reaction 2 is P, and reaction 3 will be naturally Q. We have found already 2 of them. So reaction 3 is Q. So what happens in reaction Q? Km value decreases, Vmax also changes. So it is the case of an uncompetitive inhibitor. Uncompetitive inhibitor also will bind to site other than the substrate binding site. Vmax value it will decrease, and Km value also decreases. Both changes. So it is the case of an uncompetitive inhibitor. So reaction 3 is Q. Then Km of reaction Q is less than Km of reaction R. So reaction R, I told you the Km value is 0.5. It's already given. And Km for reaction Q, that is over here, that is minus 3 point. It will be less than Km of reaction R. That is also correct. So options 2 and 4 are correct. Then CENTN1 gene encodes the protein centrin, that is localized to the centriolar apparatus. Mutation in this gene may give rise to dysfunctional cilia, leading to respiratory disorders with a different degree of severity. 50,000 individuals of a Caucasian population within a range of respiratory illness were analyzed for mutation in this gene. The red mark on the respective regions of the gene, that is over here, these are the red marks of the genes and are the position of the mutation identified. So here is the mutation that is identified. Give the results, given these results, the correct options are. So first option, a truncated protein is made, that is in case of 4. Okay, this is the fourth case. A truncated protein is made from a normally spliced mRNA, resulting in functional cilia, thereby not affecting the individual. Here no truncated protein will be made because the mutation is in the intron. It will be spliced off after RNA translation. RNA transcription. And only the exons will be joined together, so it will result in a functional cilia and thereby not affecting the individual. So option 1 is not correct. Then option 2, in case 3, that is over this one. A mutation resulted in a stop codon resulting in a partially functional cilia and hence individual have mild illness. So we can see that the mutation is near to the terminator region. Since the mutation is in the exon 2 region, it will result in an almost full protein with termination before a fully functional protein is synthesized. Why? Because the mutation is over this region. It is close to the terminator. That means after translating the exon 2 region, it will end in termination of the fully functional protein. But since there is a mutation over here, that is even before exon 2 is fully translated, it will be terminated because of that particular mutation. Hence the resulting protein is only partially functional and hence individual have mild illness. Then in case of 2, an intact mutated, that is over here, an intact mutated or truncated protein is produced resulting in dysfunctional cilia and individual with severe illness. So the mutation is just after the promoter sequence, or I can say at the start of the amino acid sequence. The mutation can over here, that is in the case of exon 1. So the mutation can be either a deletion, substitution, insertion resulting in a mutated protein being produced, or it can be even a truncated protein resulting from nonsense mutation. So option 3 is also correct. That is, an intact mutated or truncated protein is produced resulting in dysfunctional cilia and individuals with severe illness. Then option 4. The gene is not transcribed and no protein is produced resulting in dysfunctional cilia and individual with severe illness. So mutation is there in the promoter region itself. So no RNA polymerase can bind to it and the result is no transcription and translation of the gene. So no protein is produced. So it will result in very severe illness. So option 4 is also correct. So 2, 3 and 4 are correct. Then next one. During evolution, the mitochondrial genome lost many genes and some of these got incorporated into the nuclear genome. Proteins synthesized in the cytoplasm are transported to various destinations, including some that are secreted outside of the cell. Based on the transport and localization proteins within cell, the correct options is or are. So proteins secreted outside the cell are not transported via the endoplasmic reticulum. In mammalian cell, most proteins are transferred into the endoplasmic reticulum while they are being translated on membrane-bound ribosomes. So you cannot say that they are not transported via endoplasmic reticulum. So 1 is wrong. Then next one. All proteins with mitochondrial function are synthesized inside the mitochondria. It's already given that during evolution, mitochondrial genome lost many genes and some got incorporated into the nuclear genome. Or you can say that mitochondrial proteins are made in the eukaryotic nucleus outside the mitochondria. So option 2 is also not correct. Then all the proteins having nuclear function are synthesized in the cytoplasm. So RNA molecules which are made in the nucleus and ribosomal subunits which are assembled there are exported to the cytosol, while all the proteins that function in the nucleus are synthesized in the cytosol and are then imported to nucleus. So option 3 is correct. Then some nuclear encoded proteins are transported to mitochondria, that I already explained here. So option 4 is also correct. Then next one. Escherichia coli grown in liquid nutrient medium is infected with a virus at a concentration that will result in the entry of a single virus per bacterium. This eventually results in lysis or death of Escherichia coli cell, releasing the viral progeny that can in turn infect neighboring cells. Such a culture is incubated at 37 degrees Celsius and the cell-free supernatant is sampled every few minutes in order to enumerate the viral progeny. At each time point, the sampled culture is placed on solid nutrient medium while a loan of Escherichia coli, with a loan of Escherichia coli. Each virus multiplies locally, giving rise to a colony observed as clear zones or plaques and measured as the number of plaque forming units. So here Escherichia coli, it is grown in liquid nutrient medium and is infected with virus at concentrations that will allow the entry of virus per bacterium. So virus is infecting Escherichia coli, and this eventually lead to lysis of or death of Escherichia coli, releasing the viral progeny. So this culture is incubated at 37 degrees Celsius and the cell-free supernatant is sampled every few minutes. So every few minutes the cell-free supernatant is sampled in order to enumerate the viral progeny. Then at each time, and after that what they do, at each time the sampled culture is placed on a solid nutrient medium with a loan of Escherichia coli cells, and each virus multiplies locally, giving rise to a colony observed as clear zone. So they have taken the cell-free supernatant and it is sampled every few minutes and according to enumerate viral progeny, and at each time also this sampled culture it is placed on solid nutrient medium, that is this medium, that is on a Petri plate, and which is being coated with a lawn of Escherichia coli. So and this each virus multiplies locally, and in this particular plate each virus multiplies locally, giving rise to colony observed as clear zones or plaques and measured as the number of plaque forming units or PFU. So analyze the graph obtained from this experiment and choose the correct options. So the initial lag period is the time taken by the virus to attach and enter the bacterial cell. It is correct because only after entry of virus into bacterium it will result in the lysis of E. coli cell, releasing viral progeny. So at initial stage there is a lag period for the virus to enter the bacterial cell. Then an antibiotic effective against Escherichia coli when added at 0.2 hour will not result in any viral progeny. So at this particular stage, less than 1, time is 1 hour over here, so even at that is during the lag phase itself. So an antibiotic effective against E. coli will result in death of E. coli cells at 0.2 hours. With the absence of E. coli cell, virus cannot multiply. So it will not result in any viral progeny. So option 2 is correct. Then next one. When Escherichia coli is infected with an attenuated virus, a similar sigmoidal curve should be observed. In attenuated virus, the virulence of the virus is reduced and it will be viable. So it will not result in a similar sigmoidal curve. So virulence of the virus is reduced and it will not be resulting in the same sigmoidal curve. Then the slope of this sigmoidal curve is not indicative of rate of viral multiplication. The slope indicates a burst, that is host cell release many virus particles, that is assembly and release. So option 4 is also not correct. So that's it. Please do like, share and subscribe, and thank you for watching my channel.